talk about networks, I talk about systems that consist of nodes and links and that are models of connections or models of interactions. The basic elements of networks are nodes, and the nodes are connected by links. Links may be unweighted, meaning that every connection in the network is sort of equally strong, equally important, or then they may have a weight, meaning that some connections are stronger than others. Weights may have direction, meaning that we are able to travel from A to B, but maybe not back from B to A, or they can be non-directed. And when I talk about functional brain networks, I practically talk about networks that are weighted, but not directed. The background image on this slide, by the way, is a photo of an artwork by Thomas Saraceno, Algorithm. If you ever have a possibility to visit a Thomas Saraceno exhibition, please do. He's great. He's making art out of spider webs and radio wave frequencies and network models and whatnot. And when we think about modeling a system as a network, there are two questions we need to consider. To this end, I'd like to introduce 2D space. We are getting back to this space later on. The two questions are, first, on the y-axis, is our system actually a network? And by being a network, I mean the system essentially consists of nodes and links. So fundamentally that being a system of nodes and links is the very nature of our system, and we can't imagine the system not consisting of nodes and links. So this sort of like philosophical question, is our system a system of nodes and links? And then on the x-axis, another question, should our system be studied as a network? And by this I mean, does the network model give us new information? Does the network model has useful applications on our system? These two questions are at least partially independent. You can have a system that is very fundamentally network, but actually network theory can't give you much useful information. And way more commonly, other way around, you might have a system that is maybe not really fundamentally a network, but you can still get a lot of useful information using the network model. As said, I'll come back to this space in the context of brain soon. But first, a couple of words about why would we like to model the brain as a network. Now, this is actually a bit of repetition from the talk by Markus. Practically, what we know is that at the level of cells, our brains, the brains of everybody, consist of neurons that are connected by synapses. And the picture here is from a paper by folks in Madrid in the Biological Networks Lab. What they do there is that they actually have neurons of grasshoppers. They have giant grasshoppers and they have really big neurons. So it's possible to take a slice of their brain and actually image the neurons with a normal light microscope. And that's what they did. They practically destroyed all connections between the neurons and then took light microscope images several times a day to see what happens. So in the first fig, we see that there are the neurons without no connections, but in a couple of days, they start build new connections. And at around day 6, those neurons that are not connected start to die out. So there's something really fundamental in the networkness of neurons. They need to be connected. If they are not, they die out. And in around 12 days, you are able to see a complex network structure with hubs and clusters and hierarchy. 12 days is around the time you can keep the grasshopper neurons alive, then they die out. So at the level of neurons, we have a strong motivation for modeling the brain as a network. And then there is, of course, further evidence coming from functional neuroimaging, from MEG, EEG, fMRI, that for most tasks, we need more than a single brain area, which means that there must be some sort of interaction, some sort of connection between the brain areas. And because of that, it feels natural to model the whole thing as a network. So that was the why part. Why would we model the brain as a network? And now we are getting back to the space. where is the brain located in the is a network, should be a network, should be studied as a network space. I take a historical perspective on this. So we start from the late 19th century. We have here Camillo Golgi and Joseph von Gelach. They were the fathers of the reticulum theory. And now the reticulum actually happens to look a bit like the slime molds. So this was before the neurons were discovered. They had the idea that the brain is somehow mesh-like materia, where we have sort of crossing tubes. So it was a grid or a mesh, but with no neurons. Maybe looked a bit like a ball of wool. And I think that is sort of thinking that, yeah, this whole thing is a network. The connections are important because the reticulum was basically only connections. But they never made any initiative to study the thing as a network. They just said that, yeah, the connections are super important, but they never studied it as a network. So that was is a network, but maybe shouldn't be studied as one. What happened? The neurons were found by Ramon Cajal. There is one of his neuron drawings, and that actually looks really a lot like a graph or like a network. So practically what he did, he introduced the possible nodes for our brain networks, and that would have been a step towards modeling the brain as a network. But it didn't really happen. Although he made those graph-like models, he never applied any network tools. Instead, what happened? Here we have Corbinian Brodmann and Cecil and Oskar Vogt and others in Berlin around year 1900. And of course, the drawings by Brodmann. What they thought was that, okay, in the brain we have neurons, we have different neurons at different parts of the brain. And probably those different neurons must be doing different things. So they went on studying the brain with an ordinary light microscope, trying to locate areas dominated by different neurons. And they never claimed that there are no connections between the areas. They never claimed that the connections don't matter, only that they didn't concentrate on the connections. And because of that, the whole neuroscience society for years and years and decades decided to concentrate on finding areas specialized in special tasks instead of finding connections between the areas. This dominated the neuroscience understanding of the brain for years. Only started to change in 80s. The first C. elegans paper came out, and that is actually a complete map of the structural network of the C. elegans neuronal system. So it took the focus back to the connections. And then there were also papers about the structural connections in cat, in fruit fly, and so on. But the big change only happened in early 21st century. There were two waves, first around 2005. Spons and others publishing the first paper mentioning connectome. Hackman on the same year publishing his thesis mentioning connectomics. And there had been the first papers mentioning brain networks a couple of years before, but that was like I'm sorry, you should have decided to use connectome. That was the sort of dawn of the new view of the brain as a network. And then the second wave came around 10 years later. The first ones to use the word network neuroscience were Bassett and Maldon in 2016. And the sort of principal paper on the topic, the one called Network Neuroscience, was by Bassett and Spons in 2017. And these two waves of seeing the brain as a network sort of located the brain very strictly in the corner where it is fundamentally a network. It consists of nodes and links, and that is the very nature of the brain. And by studying it with the network tools, we can get a lot of useful information. That is sort of where we are now. Network neuroscience. When I started my PhD in 2014, I didn't really have a name for the field of my PhD. I used to say that, yeah, I'm a network scientist interested in brain, or maybe that I'm a computational neuroscientist. And then the term network neuroscience only came a couple of years later. And then for a minute, I said that, yeah, I'm a network neuroscientist. So it's practically a field of science that applies tools from network science to study the brain. So to put, there are two aims. We want to understand how the healthy normal brain functions, and then we want to find causes and possibly treatments for diseases. The overall scales are really broad, ranging from molecules to neurons to brain areas, even to humans. There is a thing called multiple person neuroscience, where you practically investigate the connections between brains of different people, and time-wise ranging from milliseconds, what happens at the level of neurons, up to years, up to the whole human lifespan. And as Markus mentioned, there are basically 3 different types of networks in the brain. We have structural ones, where the connections actually exist. So there are actual action bundles between brain areas. Then there are the functional networks I'm concentrating on, where the connections don't really exist. So they are just co-activation. A functional connection means that 2 things are doing the same thing at the same time. That's all. And then there are the effective networks, where the connections sort of exist, meaning that something in A is causing something in B. And final word about the space, where I would sort of like to take network neuroscience, would be the bottom right corner. Moving from the idea that the brain is fundamentally a network, merely to idea that the brain might be a network or not, but in any case, we can get useful information about it using the network tools. And why I would like to go there is that there are actually a lot of problems with the network model. There is a huge problem of defining network nodes. I'm coming back to that. There is a problem of defining network links. There are problems connecting structural and functional networks. We don't exactly know how the structure explains function, or how the function formats the structure, if you want to put it that way. And then there is the big interpretational problem that people study functional brain networks, and then they say that, yeah, we found interaction, we found causation, and that never happens because they are actually just correlations, temporal similarity between A and B. But although I'm not completely sure if the brain is a network, I'm completely sure that the network model can produce useful information about the brain. As we have already today seen, there are a lot of applications, especially in the clinical area, where you can, for example, use network model to predict what is the treatment outcome. I don't want to question that. So that was the sort of more philosophical why and what part. And unless somebody has questions or comments at this point, we move on to the more concrete how part. This is the simple recipe of building a functional network from neuroimaging data. So if any of you have ever built functional networks, this is what you have done. So we have functional neuroimaging signal. This is not actual signal, this is white noise I simulated with Excel. But it could be fMRI or MHC or EEG. You do some pre-processing to get rid of noise, to increase signal-to-noise ratio, to increase signal quality. Just to mention that there are many things that can go wrong in the pre-processing. I'm not showing examples today, but you can practically ruin your outcomes. Then you need to have the definition of nodes. And that is something I'm going to talk about today. When you have the nodes, the typical thing is that the nodes are not what you measure. So you need to somehow coarse-grain your data, collapse time series together to get a time series for each of your nodes. And then when you have the time series for each of the nodes, you throw in some definition for links. In functional brain networks, the way most common is Pearson correlation coefficient. Although in MEG and EEG, especially, people use also wavelet coherence or different phase similarity measures. But the way most common is person correlation. Then you just calculate the similarity, whatever you have used for defining links between every of your nodes, and you get a similarity matrix. That is already a full weighted network. So theoretically, you could take the similarity matrix and start performing network analysis. Normally, you don't, because you only want to keep the most strong, most significant edges. So you apply some level of thresholding, somehow get rid of those links you think are insignificant or too weak or too noisy, whatever. And then you actually have a brand new, nice, fresh functional brain network and can start analyzing it. One thing that is, okay, I need to get some water for not losing my voice. One thing that is very commonly ignored when building the functional brain networks is the time. Practically, the fact that the networks change in time. It was Heraclitus, the Greek philosopher, who said that one never steps to the same river twice. The reason is that one never looks at the same network twice. The world around us is changing. Our brain needs to adapt to the world that is changing. So it's more than natural to assume that actually the networks of our brain are changing as well. However, this is really often ignored. Typically what is done is that you take the whole time series from your measurement and use that for constructing a single network, and then you assume that results from that single network generalize to all situations. However, there have been investigations on changes in functional brain networks. Changes have been reported spontaneously over time, even when subjects are resting in for example, in the fMRI scanner, and we then divide the measurement into two. We can see that there are changes between the beginning and end of the measurement. It's commonly known that the brain networks change between tasks. That's practically how people are able to report that, okay, connection between A and B is important for a certain task. They have actually detected changes in the network between tasks. We know that functional brain networks change with age. There are maturation effects when kids grow older, and then there are also effects of aging. And then, of course, they change between health and disease. We know that many diseases, for example, schizophrenia or Alzheimer's, affect the functional brain networks. Changes have been reported both in links, meaning that, okay, this connection between here and there gets stronger or gets weaker or disappears, but also in nodes. Some nodes are actually not present in some networks or at all times. And in case somebody really wants to get into temporal changes in functional brain networks, the paper by Irai and others, 2019, and the other chromectome papers, for example, papers from the Calhoun lab, they are really good. After the how part, we are now getting to my favorite, the how not part, which is practically defining nodes in brain networks. That's what I made my PhD about, how to not define the nodes, and then I made a postdoc about trying to tell how to actually define them, and we are not completely finished with that yet, but I'll show some preliminary results. So at the level of neurons, we have a very clear candidate for nodes. The neurons are the nodes. However, the human brain contains something like 10 to 11 neurons. Clearly, we can't use them as nodes. They are impossible to measure. And even if we could measure them, the size of the network would be next to impossible for analysis. So we can't use neurons. This means that in the literature, there is huge variation in how nodes are defined. Simply the number of nodes varies from less than 100. I think the sort of smallest number of nodes would be as small as 18, where the nodes are then so-called brain systems. Up to around 10 to the 5th, that would be the number of measurement points in fMRI, or also the number of source points in MEG and EEG if you do the source reconstruction. It has been known for almost 20 years now that the definition of network nodes affects the properties. Wang and others in 2009 were among the first ones to test that, okay, if we use fMRI measurement voxels or if we use larger brain areas, what happens? And they actually reported changes in observed network properties. The common strategies for defining nodes. I think there are others as well, but those three are the most common ones. One can use directly the measurement points, both in MEG, EEG, and in fMRI. One can build sort of random clumps of measurement points. That would mean selecting random seeds and growing spheres around those. Or then, and this is probably the most common at the moment, one can use so-called regions of interest, ROIs, ROIs, that are brain areas. They are collections of measurement points, voxels or vertices, defined in terms of, for example, anatomy, saying that, okay, we have this sulcus here, that cyrus there, and between them we have an area. Or in terms of cell architecture. So some of the old Brodmann areas are actually still used as ROIs. The idea is to say that, okay, in this area we have certain type of cells, so they form an area. Or they can be defined by function. In earlier measurements, we found that these measurement points do the same thing during a measurement, so we say they form an area. And I'm now leaving out the random clumps, because I have never really understood what would be the reasoning behind using them, apart from building a null model to which to compare things. So I'm more concentrating on the measurement points, and because my work has been fMRI, I'm concentrating on the fMRI voxels and then the regions of interest. The voxels, they are the fMRI imaging resolution. The signals are almost certainly known to be noisy. They are around 10 to the 5th, and this means that if you use them as nodes, this means significant, not impossible, but significant computational load. On the other hand, the ROIs, they are collections of voxels defined in different ways. Independently on how they are defined, they are assumed to be homogeneous. So the assumption is that every voxel in an ROI does the same thing at the same time. Their time series are correlated. And because of this, one is able, this is now the coarse graining part, able to collapse the voxel time series into the ROI time series by simply averaging them. This is really widely used, widely done in the field, and quite little attention is paid on can this actually be done, how violent this is, how much information we lose. So this is what I wanted to investigate in my PhD. We, to that end, that was in 2017, defined a measure for the homogeneity of ROIs. We called it spatial consistency, and it was simply the average Pearson correlation coefficient between all voxel time series in a region. So it's pretty simple to calculate. It's extremely simple to interpret. Practically, if we have spatial consistency of 1, this means that we have a perfect ROI, everything is good. We can safely average the voxel time series. If we have spatial consistency of 0, this means that our voxel time series are completely randomly picked. There is no correlation between them, and if we average, we are left with white noise, because we average out all the features unique to each voxel. We calculated spatial consistency for several commonly used parcellations. We have the Harvard-Oxford, which is still among the most commonly used brain parcellations for fMRI. We calculated for AAL, same thing. For BrainNetOme, that was then back in 2017, sort of the top-notch new multimodal parcellation. And for all of them, the distribution of spatial consistency peaks around 0.2, telling that actually all the ROIs of the commonly used parcellations are rather far away from being perfect. Or simply put, that they are actually rather bad. If you use them, you lose a lot of information. And of course, we wanted to understand what causes this, why they are so bad. Is there an issue with defining them? Could we fix this by just defining new ROIs? Maybe all the people who defined this before have missed something. That was one option. And then other option was that maybe the spatial consistency is actually fluctuating in time. Maybe there are some moments where it's really high, some where it's really low. But when we consider the whole time series at once, we only see something rather mediocre. We don't see the tops or the bottoms, but something in between. So what we did was that we divided our time series into time windows, and for each ROI, calculated voxel level correlations inside each ROI. Now these 4 rows we have here, each corresponds to an ROI. And for each of them, we have calculated the voxel correlations in 5 time windows. And what we see here, first of all, we see that the 2 top rows have overall higher levels of spatial consistency or higher levels of correlation between voxels than the bottom rows. So we can say that, okay, the top rows are ROIs with high consistency. The bottom ones are ROIs with low consistency. Then we have that third row here, where practically the correlation distribution between voxels is uniformly low in all time windows. We maybe can say that this could be a badly defined ROI. Maybe it never gets consistent, but all the other ROIs, and this was also way more common in the data, not just that I picked the interesting ROIs for the presentation. What we see is that there are moments of higher consistency, higher correlations between voxels, moments of lower correlations between voxels, and we have those modules inside areas appearing and disappearing and changing. This is something we can't really explain what is the functional meaning of those modules. I have 2 hypotheses. One of them is that when an area gets activated, when it's more involved in a task, it maybe gets more homogeneous as a sign of concentrating on something instead of idling. And then the other hypothesis that we are investigating but haven't got any convincing results about yet is that maybe there are some areas that are sort of bridge builders in the network. So they have a topological role of being bridge builders, and they need to have several internal modules in order to build connections in different directions in the network. So those would be the shortcut areas. We are working on this. But no matter what is the actual explanation of these internal modules appearing and disappearing, what this tells us is that spatial consistency changes in time, which means that most probably we simply can't define ROIs that are stable. We can't expect that we can find an area that is all the time homogeneous. If we want to have homogeneous nodes, and we won't, because otherwise we would lose data, then we actually need to define nodes that change in time. Okay, just to remind that this is currently ignored in most network construction pipelines. So how should we solve this? This is now work in progress, going on with Tarmo Nurmi, Maria Hakonen. Tarmo is a multi-layer network scientist. Maria collected the data, Iiro supervised the data collection, and Mikko Kivelä is a multi-layer network guy. So our approach is based on multi-layer networks that are networks that combine multiple types of connections into a single network. Each type of connections presented on a different layer. Typical examples would be, for example, transportation networks, where you have layers of bus lines, train lines, maybe flights, or social relationships where you have layers of friendship, kinship, enemy. In this case, our layers correspond to time windows, and connections on each layer correspond to the functional network connections in each time window. why we want to have the separate layers for separate time windows is that we optimize the ROIs based on the data separately on each layer in each time window. We do this with a 3D clustering algorithm. I'm not going into the details of the algorithm now. We have actually tested a couple of versions of it. But the idea would be to cluster the voxels so that the functional homogeneity is maximized in each layer separately. And as you can see, this is of course a schematic fig.
How does this depend on the choice of time?
Excellent question. So far, I can only say that it depends. We haven't actually tested if there is some sort of a lower limit, like what is the lowest window length with which you can still see the ROIs changing in time. I suppose there is one. So the system has some sort of like characteristic time scale, but I don't know what that is. Of course, the window length is limited by the properties of the data. You need certain because we define the links as correlation, then we need certain number of points for calculating the correlations. So far, we have time windows of 80 samples, 100 samples. That would be how much that would be in absolute time, couple of seconds. We actually investigated the effect of time window length on spatial consistency. So we calculated spatial consistency of ROIs in, practically we took the whole time series of each ROI and then started cutting samples and saw what happens to the spatial consistency. What happens there is practically that we have the spatial consistency denoted by with phi for historical reasons, and then we have the number of samples. What happens is that it first increases and then at some point saturates, and this saturation point here is around 80, and that's where we got the window length of 80 samples. And this has also been used in the literature. For example, Daniel Bassett's lab typically uses 80 samples for fMRI. But that would be investigated, actually should be investigated. My guess would be that if you have shorter windows, then the ROIs change less. So after defining the ROIs, we define the links in each layer as Pearson correlation. So we have a functional network for each layer. And then we define the links between the layers in terms of spatial overlap. So practically the interlayer links tell how much this ROI changed. If they stay stable, then we have a super strong link between each ROI and its replica on the next window, in the next window, on the next layer. If they change a lot, we have a bunch of weaker links. So part of the existing ROI is actually merged into other ROIs. For example, there you can see that if you look at the blue ROI there, it first stays unchanged between the first two windows, so there's a very strong link. And then what happens is that it almost disappears, only the seed remains. So we have a very weak link between the seeds and then stronger links from the blue one to other ROIs, showing that that's actually where the voxels from the blue ROI went. So this is the model we are building right now. And this is actually the first time ever I saw any preliminary results from this model. This is also a schematic. So what I have done here is that I have simulated data by selecting random seeds and then building a spherical ROI around each seed and defining the time series of each voxel inside each spherical ROI as a mixture of the time series of the seed and white noise, so that the similarity to the seed decays following a Gaussian function. So and then I have calculated for each voxel the similarity to its closest neighbors. So practically there you see the white areas, they are areas of very similar voxels. And then there are the red areas that are sort of the boundaries between ROIs, where the voxels are not really similar to anyone. The black lines on top of the data are ROI boundaries. The first ones are from the Brenneton parcellation. And now, of course, Brenneton fails here really badly. That's because we didn't even aim for natural-looking brain data. It was just simulated data. So it's understandable that Brenneton doesn't match it at all. But there you can, for example, see that Brenneton is... You can see that Brenneton is practically dividing a homogeneous area into two. And we assume that that happens sometimes in the real data as well. Then the optimized boundaries are actually obtained by our algorithm. And there you can see that it very nicely finds the homogeneous areas and separates them from each other. And then actually, if you repeat this for other set of simulated data, the Brenneton boundaries are exactly the same because it's a static parcellation, and the optimized boundaries change because they adapt to the data. Here I have the spatial consistency calculated for Brenneton, that's red. Random blobs of voxels, that's blue. And you can actually see that the Brenneton parcellation that is built based on anatomy and function is scarily similar to random blobs of voxels. Then we have the Craddock parcellation. That is not actually the static parcellation given by Craddock and others. So if you have some brain network building package and you have the Craddock parcellation there, that's a static parcellation. What we have applied here is actually the clustering algorithm behind the Craddock parcellation. And that's why it's doing better than Brenneton. It's adapting to the data. And the two last ones are two versions of our own algorithm that are slightly outperforming the Craddock parcellation. So this, how much we can increase the functional homogeneity by using optimized ROIs. And now that we think about this model, we are not saying that our clustering algorithm is the best. Every network scientist wants to introduce a clustering algorithm and claim that it's the best one ever. We are not really saying that. We are just saying that one can improve functional homogeneity using a clustering algorithm, and theoretically one could plug in to this model whatever clustering algorithm. We are just using one example. Now, of course, what we want to do with this model, there are a lot of open questions. Some of them are such that we are working on them right now, especially the question about how ROIs change. We already know that they do change, but we would like to be able to quantify what they actually do. Do they split? Do they merge? Do some of them disappear? And of course, we would be interested to see what is the timing of those changes. Do we have sort of states where all the ROIs stay stable and then suddenly all of them change? Or is it more a gradual thing? One of them is changing now, other one is changing in the next time. Of course, it would be also cool to see if there are those state changes. Do they happen to overlap with changes in the stimulus? But these are things we are working on. And then I would be interested to see how this whole thing is affected by diseases, and especially we would have nice data from Alzheimer's disease patients. So I would like to see how this whole thing is affected by Alzheimer's. So to conclude, network theory surely can help to understand the human brain. That was the x-axis of the space. We can get useful information about the brain. However, not all that glitters is actually a network. And the open question is the human brain actually a network? That is the y-axis. We know that network construction from functional neuroimaging data is not trivial. There is the huge problem of node definition, and then there are other huge problems I haven't covered today. For example, how to pre-process the data. how to define the links, how to threshold the similarity matrices. So whenever you go for constructing a functional brain network, know your methods and think, why I'm using these methods, are they good for building my network? And importantly, and often ignored, brain networks, especially the functional ones, they change in time. So if you include time in your analysis, that opens new horizons, allows to understand the brain function better. And one example on how to possibly do this is using time-dependent nodes. We are not the only ones developing time-dependent nodes. There are others. There was even a paper doing network analysis with time-dependent nodes, but there the nodes weren't defined for time windows, but merely for tasks, so the time scale was longer. Importantly, what I think network neuroscience needs is critical thinking, criticizing what we have now, criticizing our understanding of the brain, and discussing how to get better. Here are all the references mentioned in the slides, in case you want to get back to them. Thank you. Now it's time for questions and comments, if you have anything